Good morning everyone. Today's topic is taste and smell. So these are sensations, special senses. So the coming to taste, the primary taste sensations are sweet, salt, sour, bitter and umami. Umami has been added recently. Sweet, it is best appreciated at the tip of the tongue. Salt is best appreciated on the lateral margin of the tongue. Sour, when you eat lemon, it is sour. It is best appreciated on the lateral margin of the tongue. Bitter is appreciated at the back of the tongue. Umami, it is a pleasant, sweet taste sensation, different from standard sweet taste. It is a sensation you get, taste sensation you appreciate when you eat meat. Now, coming to taste buds. The taste buds are the receptors for taste sensation. They are present on the walls of the papilla. Taste buds are oval-shaped structures containing receptor cells, supporting cells and basal cells. In this picture you can see a taste bud where you can see gustatory cell, supporting cell and basal cell. These gustatory cells have hair-like structures which come out of the taste pore, small opening at the apex of the taste bud, and these hairs are called microvilli, and supporting cells are innervated by nerve fiber. Gustatory cells are innervated by nerve fibers. At the tip of the taste bud there is a taste pore. From the apical portion of the receptor cells, hair-like projections come out of the taste pore. These are called microvilli. The fine sensory nerve fibers which arise from the receptor cells carry the taste sensations. The basal cells form the receptor cells, that is gustatory cells, which are replaced periodically. Coming to nerve supply of the tongue, the taste sensations from anterior 2/3 of the tongue are carried by chorda tympani nerve, which is a branch of facial nerve. From posterior 1/3 of the tongue, the taste sensations are carried by glossopharyngeal nerve. Coming to taste pathway, nerve fibers from anterior and posterior parts of the tongue end on gustatory portion of nucleus tractus solitarius (NTS), which is present in the medulla of the brain. The second order neurons arise from NTS end in ventral posteromedial (VPM) nucleus of the thalamus. The third order neurons arising from thalamus end on the face area of the somatosensory cortex, lower lateral part of postcentral gyrus, deep in the lateral sulcus. That is a region of taste sensation reception in the brain. Applied aspect to the taste. Ageusia means there is no taste sensation. Absence of taste sensation is ageusia. Then when there is decreased sensation of taste, it is called hypogeusia. Dysgeusia is disturbed taste sensation. So this brings us the taste. Now we will go to smell. Smell or olfaction. The olfactory receptors are located in yellowish pigmented olfactory mucous membrane of the nasal cavity. The olfactory receptors are called olfactory cells. There are about 10 to 20 million olfactory cells. They are interspersed between the sustentacular cells, which are supporting epithelial cells. Now, olfactory cells are bipolar cells. The dendrites, that is, the cell has two poles. At one pole there is dendrite, the other pole there is axon. The dendrite has an expansion called olfactory rod. The cilia project into the mucous membrane from these rods. The axons arising from olfactory receptors pierce the cribriform plate of the ethmoid bone and enter the olfactory bulb. The olfactory neurons are the only neurons known to be replenished by cell division. Mucus is produced by Bowman's glands. Olfactory pathway. The axons of the olfactory receptors pierce the cribriform plate of ethmoid bone and end in olfactory bulb. The short axons of the olfactory cells terminate in multiple globular structures within the olfactory bulb called glomeruli. Each glomerulus has dendrites from mitral cells and tufted cells. So these are second order neurons. Mitral cells and tufted cells are second order neurons. These cells in turn send axons through the olfactory tract to transmit the olfactory sensation to CNS. So in this picture you can see the olfactory membrane, that is down L1 mucous membrane, where orange cells are the olfactory receptors and the pink cells are sustentacular cells, that is supporting cells, and then these olfactory receptor is a bipolar cell. One end bipolar, it has two poles. One pole or one end is the rod-like structure with hairs, and the other end has nerve fibers. These nerve fibers pierce the cribriform plate of ethmoid bone, that is L1, that is bone, and enter the CNS, and they reach the olfactory bulb. In the olfactory bulb, they end in olfactory glomeruli. These olfactory glomeruli receive dendrites of mitral and tufted cells. Mitral and tufted cells are the second order neurons, and the axons of mitral and tufted cells form olfactory tract. From the olfactory tract, the fibers go to the olfactory nuclei in the CNS. The olfactory tract enters the brain at the anterior junction between mesencephalon and the cerebrum. The tract divides into two pathways. One passes medially to the medial olfactory area and the other laterally to lateral olfactory area. The medial olfactory area consists of a group of nuclei located in the brain. Lateral olfactory area is composed of piriform cortex amygdaloid nuclei. From here, signals pass to limbic system. So, these will help in perception of smell. Now, coming to applied aspect, anosmia means loss of smell sensation. Parosmia means perverted smell sensation, that is, you will not feel the exact smell, but it is different smell. Thank you. This finishes the special senses.